Okay, so this is my last lecture, right, for extension topic A. We're going to look at bacterial diseases and antibiotics. So before we start, just a reminder that under the extension topic A, there's also the viral diseases, right? So that part has been done under the virus lecture. Okay, so please refer to that virus notes and virus lecture, right, to complete the extension topic A for the viral diseases. Okay, so just looking at the learning outcomes covered in this lecture. Okay, we're going to look at the bacterial diseases with the tuberculosis by the bacterium, right, Mycobacterium tuberculosis as an example. And also looking at the antibiotics with penicillin as an example. Okay, so for tuberculosis, okay, so for the general bacterial diseases, right, some of the common modes of transmission of different bacteria. Okay, I'll leave you to read the notes yourself. Okay, so we just go straight to the example of tuberculosis, or TB for short. Okay, so the pathogen is the bacterium, right, Mycobacterium tuberculosis, or sometimes you might see it referred to as Mycobacterium tuberculosis bacilli. Right, bacilli is just to describe the shape. Right, the shape is this rod shape, as you can see in the slide. Right, this rod shaped kind of bacterium. Right, so that's known as bacilli. Okay, so sometimes it's called Mycobacterium tuberculosis bacilli. Okay, so anyway, the bacterium is spread via the airborne transmission, so that would involve the small respiratory droplets, or also called droplet nuclei. So these are very small droplets produced when we sneeze, when we cough, or even talking, singing. All right, so when we do that, we will produce different sizes of respiratory droplets. For those that are smaller, that are able to travel for a longer distance and able to remain suspended in the air for longer. Right? So all these are known as small respiratory droplets, and for such transmission is known as the airborne transmission. Okay, so if you have read the front part of the notes, right, then you would have looked at the different transmissions modes for the different bacteria. Right, so we have droplet transmission versus airborne transmission. The difference really is in terms of the size of the droplets, how far the droplets travel, and how long the droplet stays in the air. Right, so for TB, it is referring to the small ones, right, the small droplets that can travel longer and stay longer in the air. Okay, so for the patient that have got the TB bacterium right inside his lungs, to be able to cough out or sneeze out the droplets containing the bacterium, right, this patient would have active TB. Okay, right, so when the person, the uninfected person, breathed in the droplets with the bacteria of course will get breathed into the lungs and into the alveoli. That's where there are the presence of macrophages, right? So we call them alveolar macrophages, just to specify the location. They are simply normal macrophages, not too different from those in the blood or whatnot. Okay, so anyway, these macrophages would phagocytose the bacteria as well as the dendritic cells present. Right, so that's the usual innate immune response in the presence of a pathogen. But the bacteria, the Mycobacterium tuberculosis, has evolved a way to block the fusion of the lysosome to the phagosome when it is phagocytosed by the macrophages. Okay, so the diagram here is a bit chim, right? It shows you the different mechanisms, different pathways, how the bacterium is able to block the fusion of lysosome to phagosome. Because you don't have to know all this mechanism. All you have to know is the end result, which is shown by the cross here, that the lysosome doesn't fuse with the phagosome to form the phagolysosome, right? This doesn't happen with the Mycobacterium tuberculosis in the macrophages. Okay, so what happened is the Mycotuberculosis, the Mycobacterium tuberculosis, right, is able to not only block the killing of itself by the lysosome, it is also able to then escape from the phagosome and get into the cytoplasm of the macrophages, and it will replicate inside the macrophage, right? So the macrophage becomes infected and eventually it will die. So the macrophage dying, right, due to this infection by the Mycobacterium tuberculosis, it is described as macrophage necrosis. Necrosis just means cell death. So the dying of the macrophages. Okay? And when the macrophages die, then of course the replicated, right, bacteria would be released. And being bacteria, right, they're not viruses. They don't absolutely require a whole cell. They are alive on their own, right? They are bacteria. So they are able to also replicate outside of the macrophage as well. Right? So whether it is inside the macrophage after the failed attempt at killing them via phagocytosis, or after the macrophages die and the bacteria are released, the bacteria just keep dividing. Okay, and when the bacteria get recognized by another macrophage and get phagocytosed, then this whole process repeats again, right? There will be no killing, there will be replication inside the macrophages until the macrophages die, right? And then the bacteria get released and they keep dividing again. Okay, so this is what happened with the bacteria. So our immune system is not entirely helpless, even though the macrophages are not really able to kill the bacteria, right? But our immune system is not helpless against this bacterial infection. So what our immune system does is, right, if you recall from the innate immune response, macrophages, besides phagocytosis, they also release inflammatory cytokines, right? Cytokines that result in inflammation. Okay, so one of the effects of the cytokines is to recruit more of the other immune cells to the site of infection, right? It may be recruiting more macrophages. It can be recruiting, say, neutrophils, right? And other immune cells. Okay? And also, right, remember there's also the dendritic cells that would have phagocytosed the Mycobacterium tuberculosis. So they are a bit more successful in terms of breaking down the bacteria. So they will be able to present the fragments of the antigens, right, to the specific T cells, be it helper T cells or cytotoxic T cells that recognize the presented antigens. Right, so they will activate those T cells, and once activated, the T cells will then migrate to the lungs, to the alveoli where the infection occurred. All right, so with all these cells that are recruited, that are migrating to the site of infection, they will then surround all these infected macrophages. The cytotoxic T cells will try to kill the infected macrophages, so that would limit at least a bit the replication of the bacteria, even though the bacteria doesn't really require the whole cells, right? So they can still replicate outside of the whole cells, but killing the infected macrophages can still limit a bit the replication of the bacteria. But what's more important is the accumulation of all these different immune cells will be able to form a structure called a granuloma or tubercle, which is kind of shown here, and a more mature one here, right, is formed by a layer up. This is showing one layer. It should be layers upon layers of immune cells that are surrounding the infected macrophages as well as the released bacteria. All right, so what the granuloma, so it's called granuloma or tubercle, right? What the granuloma or tubercle does is it blocks, right? It contains the bacteria from spreading to other parts of the lungs, right? So all the bacteria are contained via this physical blocking by the immune cells, right, within this granuloma. So within the granuloma, right, the bacteria may still continue to divide, but after a while they would turn dormant, right? So they would stop dividing, they would just persist within the granuloma. So when they are dormant, right, the immune cells also will not really touch them, or I don't say will touch, right? It wouldn't really disturb them, right? So when they're dormant, they are kind of evading the immune cells that are in the granuloma, right? So the immune cells don't really kill them, but the bacteria also don't really do anything, right? So they are dormant. So this is when we have a latent TB infection, right? So all the bacteria are contained within the granulomas, right? So there's no spreading of the bacteria to other parts of the lungs or even to other organs. There's also no transmission, right, to other people, because when the patient or the infected person cough, but there's no bacteria in the respiratory droplets because the bacteria are all contained, right, within the granuloma. So there's just latent TB. The infected person is as normal as he or she can be, right? No symptoms whatsoever. All right, but when the immune system is weakened for some reasons, right, be it because the infected person contracted HIV, or maybe the infected person had a organ transplant and was taking some immunosuppressant drugs, for example, right, then the immune system or the immune cells, right, would then be able to contain the bacteria anymore within the granuloma. Okay, so that's when the bacteria can escape from the granuloma, and they will no longer be dormant. They will be reactivated again, and they can then spread to other parts of the lungs, replicate rapidly to overwhelm the weakened immune system, and spread to other parts of the body, to other organs, and also get transmitted, right, to other people, right, because they can now be found in the respiratory droplets when the infected person sneezes or coughs. Okay, so that's when it transits to active TB, and this happened to about 10% of people that get infected with TB. So most people will just remain, or most people will just have latent TB, but about 10%, right, will be able to develop this active TB. Okay, if not treated, of course. If someone has received treatment even during the latent stage, then of course that's not going to be the switch to active TB, right, because treatment has been provided. All right, so that's in a very summarized version. I hope you understand the progression of the TB infection. All right, so next we'll just very quickly go on to the antibiotics. So again, for the general stuff, right, the different modes of actions of different antibiotics, right, I'll just leave you to read the notes yourself. Right, we'll go straight to the example of penicillin very quickly. All right, so here you first of all need to recall the structure of the peptidoglycan cell wall of the bacteria. Okay, so the peptidoglycan cell wall is formed by a mixture of sugar and amino acids, right? So they are called the two molecules called NAG and NAM, which really are sugars with attached amino acids. So the sugars are the hexagons and the amino acids are those that are circles, right? And within the peptidoglycan, there are cross linkages, right, between chains of these NAGs and NAMs. Okay, so the cross linkages are actually formed by amino acids being joined together, right, between the different chains, as you can see in this diagram. And these cross linkages are formed by this enzyme, DD transpeptidase. Okay, so what penicillin does is that it actually would inhibit DD transpeptidase, so the cross linkages between different chains are not formed, as you can see here, right? This is the cross linkage between the two chains, right? So this cross linkage is not formed, or is being formed now by the active DD transpeptidase, but with the binding and inhibition of penicillin, then this enzyme wouldn't be able to form this cross linkage. Okay, so without the cross linkage, then obviously the peptidoglycan cell wall is weakened, right? There's less tensile strength, and the new cell wall that is formed would be weakened, right? So the cross linkage would have already been formed in existing cell wall, right, before the introduction of penicillin. Okay, so the existing cell wall is really not affected so much, right? It's the newly formed cell wall that's going to be weaker after the introduction of the penicillin, because those newly formed cell walls wouldn't have the cross linkages, right? So penicillin really affects the daughter bacterial cells after binary fission, because the daughter cells have the new cell wall, right, that are weaker, right? So then the daughter cells with the weaker cell walls, right, would lyse and die, right, under the osmotic pressure, right? Water will enter the cell. These cell walls are not strong enough to prevent bursting of the cell, right? So the cells burst and lyse, and therefore the daughter bacterial cells die. So that's how penicillin causes the death of bacteria. Okay, so that's just one slide on penicillin, very fast one. Okay, so that's all for this lecture.